Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
You know how we often think of mitochondria as just the cell's energy factories, quietly buzzing along, generating power? Well, what we're diving into today might just flip that whole understanding on its head. Get ready for an aha moment, because groundbreaking new research reveals they're not just passive powerhouses, they're actually secret active defenders.
Active defenders.
Capable of starving, invading pathogens right within our own cells. It's like discovering your unassuming librarian neighbor secretly moonlights as a master martial artist.
Exactly.
Yeah.
Always ready to protect the neighborhood.
Okay, so let's unpack this. We've got some absolutely fascinating source material today, a deep dive into a game-changing 2025 science research article. Our mission is to extract the most important nuggets of knowledge about how our cells, specifically our mitochondria, fight back against unwelcome guests, specifically the parasite Toxoplasma gondii. This isn't just about understanding a new cellular pathway. It's about seeing our internal defenses with, well, fresh eyes.
Absolutely. And what's fascinating here is that for a long time, we've known intracellular parasites like Toxoplasma are incredibly cunning. They essentially hijack our cells, right? Creating their own little safe haven inside, siphoning off nutrients to copy their genomes and multiply.
Taking what they need.
Exactly. And we also know that mitochondria, beyond just making APP, are crucial biosynthesis hubs. They use these intricate metabolic pathways like the folate-dependent one-carbon pathways, which are essentially molecular assembly lines handling tiny chemical groups, critical for making vital nucleotides, the building blocks for our own DNA and RNA.
Right, the A, T, C, G, S, well, U for RNA.
Precisely. But here's the profound knowledge gap this study really addresses. Were our mitochondria merely passive resource pools for these parasites? Or could they actively, reshape their own metabolism to deny pathogens these key precursors?
Could they be more than just victims?
That was the big question. This research connects stress-activated signaling to a truly remarkable and honestly previously unappreciated defense mechanism.
That's a crucial distinction, isn't it? So moving from a passive victim to an active strategist. And this brings us to the core question the researchers then tackled. How exactly do our cells initiate this metabolic counterattack? And can they truly throttle parasite growth by simply reallocating nutrients rather than just, directly attacking the pathogen with brute force?
Yeah, it's a different kind of fight.
And it seems that folate, a vital nutrient, turned out to be the perfect battlefield for this cellular metabolic warfare.
Indeed. Folate is absolutely central to making essential building blocks like thymidylate, TTFP. That's one of the four crucial components, the TT in the DNA alphabet, basically.
Yeah.
If a cell or a parasite inside it can't make enough DTMP, it simply can't replicate its DNA.
So it can't multiply. Game over, potentially.
Exactly. And this folate is carefully partitioned between different compartments of the cell, the mitochondrial pool and the cytosolic pool. So this paper proposes a groundbreaking concept: nutritional immunity.
Right, like withholding iron systemically.
But not on that usual systemic scale. This is happening at an organelle level, right inside our cells. The idea is that mitochondrial stress signals, perhaps directly triggered by the infection itself, activate a key player, a transcription factor called ATF4.
ATF4, okay, I've heard of that in stress responses.
Yes, it's a known stress sensor.
Now this is where it gets really interesting for me. The hypothesis then was that this ATF4 activation could crank up the mitochondrial one-carbon flux, essentially creating a folate sink inside the mitochondria.
That's the term they use, yeah, a folate sink.
Imagine a powerful, super efficient vacuum cleaner suddenly activating inside your cell, sucking up all the essential Legos, those folate molecules, and hoarding them for itself.
Leaving none for the invader.
Leaving absolutely none for the invading alien to build its spaceship, or in this case, its DNA. It's a beautifully elegant strategy of deprivation.
Precisely. And the study laid out the mechanistic steps with impressive clarity. First, they observed that infection by intracellular pathogens like Toxoplasma doesn't just happen quietly. It induces very specific mitochondrial stress signatures within the host cell, which then crucially activates that ATF4.
So the threat is detected, alarm signal goes off, triggers the stress sensor. Got it.
Exactly. Then APF4 acts like a highly specialized conductor, orchestrating a cellular response. It upregulates genes that specifically drive those mitochondrial folate pathways.
Telling the mitochondrial assembly lines to work overtime.
Right. But it doesn't stop there. ATF4 also boosts mitochondrial DNA biogenesis.
Making more mitochondrial DNA, why?
Well, think about it. Making more DNA also requires folate building blocks. So both of these actions, increasing the folate pathway activity and making more mitochondrial DNA, significantly increase the local demand for folate right inside the mitochondria.
Ah, so it doubles down on sucking up the folate.
It creates this powerful internal demand. And this active reallocation, this purposeful hoarding of folate within the host cell mitochondria essentially deprives Toxoplasma of the very folate it desperately needs for its own DTMP synthesis.
Right.
Without those critical building blocks, the parasite's ability to replicate is critically slowed down, or maybe even halted. It's a resource war, and the host cell is turning the tide by controlling the supply lines.
So it's not just passive depletion where the parasite simply outcompetes. It's an active, programmed defense, a deliberate strategy by our own cells.
That's the key takeaway.
And what's truly compelling is how they proved this. They showed that if you mess with ATF4 signaling, or if you disable these specific mitochondrial one-carbon enzymes,
Yeah, if you knock down those key players.
that powerful anti-parasite effect is significantly diminished or even gone.
Correct.
Conversely, if you somehow boost that mitochondrial folate flux, you actually enhance the restriction of the parasite.
That's the clincher.
That's tangible, elegant evidence directly connecting these transcriptional stress programs, these cellular alarms and responses to actual measurable nutrient competition that impacts pathogen growth. It's hard proof that our mitochondria are active players, not just bystanders.
It really shifts the paradigm.
It truly shifts our perspective on cellular defense, doesn't it? It goes beyond simply understanding how our cells react to stress. It shows them actively manipulating their internal environment to fight off intruders.
The conceptual novelty here is absolutely huge. It transforms our view of mitochondrial one-carbon metabolism from just a fundamental metabolic pathway, a simple set of chemical reactions.
Into an active, programmable defense lever.
Exactly. It reframes mitochondria from being mere passive resource pools or power generators into sophisticated innate defense organelles with a strategic role in immunity.
This is a profound game changer for several reasons. Instead of only targeting pathogen enzymes with traditional antimicrobial,
Which often leads to resistance.
right, the pathogens mutate, the drug stops working. But this work points towards an entirely different approach. Host-directed therapies.
Mm-hmm.
We're talking about tweaking our own cell's metabolism, essentially, optimizing our internal defenses to disadvantage the pathogen.
This could potentially sidestep the resistance problem entirely because we're not targeting a mutable pathogen protein. We're enhancing our own inherent cellular strategies.
Imagine the possibilities for long-term treatment.
Yeah, and if we connect this to the bigger picture, this sort of zoomed-in model of a folate sink, it complements classical nutritional immunity, like the iron sequestration you mentioned earlier.
Where our bodies hide iron from bacteria systemically.
Right. But this folate sink operates at a much finer, more granular scale, the organelle level. It leverages a specific and absolutely crucial resource for DNA synthesis. And the sheer elegance of this approach lies in its potential for resistance proofing. The target is the host's innate ability to reallocate resources, not a parasite enzyme that can easily mutate and render a drug useless.
Which makes therapies inspired by this potentially much more robust.
Exactly. Much more durable, perhaps.
Okay, so this study focused on Toxoplasma in cell cultures, which is a powerful model, very well established. But looking ahead then, what are the immediate implications for future research? Where do we go from here?
Absolutely. Well, the limitations of any single study, like the model specificity here, always pave the way for exciting next steps and broader applications. Researchers can now rigorously test if other intracellular pathogens trigger similar ATF4 mitochondrial programs.
Like what? What other bugs live inside our cells?
Well, for instance, pathogens like chlamydia, which also heavily rely on host cell metabolism, or the insidious liver stage of Plasmodium, the parasite that causes malaria.
Right, which spends a crucial phase hidden within our liver cells.
Exactly. Those are prime candidates for investigation. If this mitochondrial defense is a general principle, applicable across a wider range of intracellular infections,
That would be huge.
it could unlock entirely new treatment strategies for a vast array of diseases that currently pose significant global health challenges.
And beyond looking at other pathogens.
There's also fascinating potential to explore small molecules. Could we identify or design compounds that could transiently bias mitochondrial folate flux?
Sort of nudge our cells towards this defensive state.
Precisely. Without causing systemic folate deficiency in patients, which would be bad. That would be the holy grail of a host-directed therapy.
A fine-tuned approach.
And finally, defining the specific tissue contexts, like in specialized immune cells, such as macrophages, or critical organs like neurons, where this defense is strongest and most relevant, could be crucial. This mechanism really could inspire a completely new class of host-biased anti-infective strategies, moving beyond the traditional arms race with pathogens.
Wow. This deep dive has truly given us a whole new appreciation for our cellular powerhouses. They are not just the energy generators we learned about in biology class, they are sophisticated strategists, actively defending us from within by cleverly hoarding and reallocating essential nutrients.
It fundamentally shifts how we understand cellular immunity and opens new pathways for fighting infectious diseases that have long plagued humanity. It's really exciting work.
And for you, consider this. How many other seemingly passive cellular components, those quiet workhorses of our biology, might be quietly orchestrating intricate defenses or playing other hidden heroic roles we are only just beginning to uncover?
Good question.
What other housekeeping functions might actually be hidden acts of cellular heroism, constantly working to keep us safe? Something to mull over next time you think about your mitochondria or indeed any part of your own incredible body.